Well, last lecture I identified it as lecture 8A. I didn't realize, but I kind of got carried away and it ran for longer and it turned into something that basically combined the content of lectures 8A and 8B together, right? 8A was supposed to be diffusion, facilitated diffusion. 8B was supposed to be osmosis. And I don't know, I guess in my mind, diffusion and osmosis are so closely tied together, it's really kind of hard for me to talk about one without going through both of them, at least in the same lecture. And so rather than recutting the video, I figured that, hey, there's nothing wrong with having diffusion and osmosis together in the same little bit of content. I could split them up, but they kind of fit together. They kind of go nicely together. And so that's how you ended up with the extra long video for the first one. Okay, but realize that's actually 2 videos worth of content, 8A and 8B, right? But as I'm looking at the content goals that I had for 8B, there are a few things that, that were mentioned in that first video, but I want to kind of tidy them up because I don't think I did enough justice to them as we were going through that content. And so what I'm going to do is to talk to you briefly about these concepts that are related to osmosis: isotonic, hypotonic, and hypertonic. Okay? And so a couple of things you should know just generally. Hypo, like a hypodermic syringe, means below. Right? Hyper, like a hyperactive child, means above. Right? You can be hypo, below, hyper, above. Those are 2 Latin prefixes that you should be familiar with. Iso, like an isosceles triangle, means equal. With an isosceles triangle, you have 2 equal sides. Okay, it's not equilateral, but it's isosceles if you've got 2 equal sides. Okay, so equal, right? And so what we're kind of referring to here is the tonicity. And so tonic here refers to the concentration of solutes with regard to osmosis. And so these are all relating to osmosis. And if you recall back from the previous video, there was an example involving a red blood cell, right? And so I've drawn a picture of a red blood cell here, also known as an erythrocyte, meaning a red cell. Erythrocytes have this very special shape. If you looked at this from the side, okay, it would have 2 thicker ends, and the middle portion would be kind of like an indented. It would be indented on both sides. It kind of looks like a flattened disc with a little bit of, it's kind of pinched in the middle, right? And so that pinched middle portion is what you have here. It's actually the spot where the nucleus of the cell once was. I don't know if you know this, but erythrocytes are different from most cells in that they don't have a nucleus. The nucleus actually was once sitting in this spot right here, but before the erythrocyte was matured, okay, the nucleus was removed. We basically take the nucleus out, recycle the materials, and make more erythrocytes. And so what actually gets left after you remove the nucleus is simply a sack of stuff, right? And the stuff that's inside of a red blood cell is the crucial bit. There's tons and tons and tons of a molecule called hemoglobin, which is a protein. It's a protein that has a quaternary structure. Okay, real quick, what does that mean? Quaternary structure of a protein is, oh yeah, that's when you've got multiple polypeptides that come together. Okay, and so hemoglobin, which is the protein that's there, super important as far as the functions of red blood cells go. It's there in huge abundances, and there are a few other proteins. But the idea here is that inside the cell, you've got lots of osmotic solutes, meaning that they're not permeating. Permeating, which means that they're not able to diffuse across the plasma membrane of the erythrocyte, so they have to stay put. Okay, and so one of these important solutes is hemoglobin. So this would be including hemoglobin, real important protein for most of you, especially those who actually are going to follow through with a nursing career or a career in a health-related field, right? Okay, that's the background. And so in this experiment that we run in physiology, you can take the erythrocyte, you could put it into a seawater, you could put it into fresh water, or you could put it into contact lens solution, isotonic saline. Okay. And the operative words here are A, hypertonic, fresh water is hypotonic. Physiological saline, the saline that you have for contact lenses, is isotonic, meaning that we have the same material, seawater, the same solute is NaCl for that. The ionically bonded material, the sodium is in the form of sodium ions, chloride is in the form of chloride ions, and the solute, sodium chloride, is either there at around 3.5%, and that is 3.5 grams of salt in 100 grams of seawater, right? So 3.5% means the same thing. Isotonic saline is at about 0.9%. In other words, 9/10 of a gram of salt in 100 grams of isotonic saline. And hypotonic fresh water is not salty at all. There might be a very, very tiny amount of salt, but it's going to be close to 0% NaCl. Okay. And basically what we're saying here is that in hypertonic, this is too high. Of a concentration of salt. In hypotonic, this is too low of a concentration of salt. Whereas in the isotonic saline, we've reached that Goldilocks position where we're just right. Right. And because we're just right, or just right means that we match the osmotic concentration. Inside the cell. Okay. And so because the concentration of isotonic saline, the concentration of salt, NaCl, outside the cell is having the equal, is creating the same demand for water molecules as what we have inside the cell. Might have different solutes there, might have hemoglobin inside the cell. But because the concentrations are, the concentrations actually are quite different, right? Because we've got high concentration of hemoglobin inside, no hemoglobin outside, a high concentration of sodium chloride outside, and a very low concentration inside. But the overall concentration in both directions is the same. So you might have end osmosis, osmotic, water moving into the cell, water moving out of the cell through exosmosis, but the two things balance. And that's how we end up with the cell being perfectly happy, right? We're not having a net influx of water. We're not having a net efflux of water. It'll be about the same, right? And so when this happens, these cells in isotonic saline are going to be ending up perfectly happy, right? They stay round, they stay inflated. They're not swelling up. They're not shrinking down. Happy cells. In other words, if you want to keep red blood cells alive, one thing you're not going to do is to put it into a hypertonic or hypotonic environment. You need it in an isotonic environment. Okay. However, if you put those same cells into hypertonic seawater, now the concentration outside is much greater than inside. And so in hypertonic seawater, you would have lots of exosmosis. Okay? And that would result in the cells losing volume because the water is leaving. And those cells will basically shrivel up into little raisins. Right? Now, in the case of hypotonic freshwater, remember the concentration outside is practically nothing. Concentration inside is high, and so you'd have movement of water into the cell, which basically increases the volume. The volume goes up until it reaches a point where the pressure is so great, it causes the cell to explode, right? So that's like the debris left over by exploded cells. Call this hemolysis. Good word for it. Hemo, this is relating to blood. Lysis means splitting apart or breaking apart. And so, yeah, if you take your red blood cells, put them into fresh water without any salt at all, they will explode. Right? You take those same cells, put them into contact lens solution, look at them through the microscope. Hey, those look just like red blood cells. Take the same cells and move them into salty water, they shrivel up like little red raisins, right? And so this was depicted for you in the video, in the last video. I think it's in one of the textbook chapters in the last video. I just wanted to set the record straight and make sure that it gave you a full explanation for what's going on with that particular vignette in the previous video. Okay, so let's go back to our content today. Okay, last time we went through this this idea, which says that if you have either diffusion, remember what diffusion is? Simple diffusion means we're moving a solute from an area where it's at a higher concentration to an area where it's at lower concentration, heading towards that equilibrium where the concentrations are equal throughout the entire system. You could have that happening. You could have the movement of the water molecules in the opposite direction. We call that osmosis. Okay? Or if you have a membrane and the membrane prevents the simple diffusion of a solute because it's large and polar or charged. Okay? These are things that will not simply diffuse across the membrane. They have to be facilitated. They have to be allowed through a specific channel. Okay? If that's happening, these were all examples of exergonic reactions because they did not require an energy source. Okay? They happen for free. And that's where we left things off. I previewed the concept of active transport, which is distinct from these forms of movement. Movement of solutes, movement of water, movement of solutes. Active transport is a movement of solutes, but it's going in the opposite direction of the concentration gradient. Right? So this would be like moving solute. Okay, so here's my membrane. Okay, and maybe we have a high concentration of sugar inside because we're storing sugars, because maybe we're an orange, right? And we might have a lower sugar outside, right? And if we're actually moving the sugars, I've drawn it deliberately so the sugar molecules are at a lower concentration on the right, higher concentration on the left. If we're moving the sugars through some type of membrane protein, right? Sugar molecule out here can be scooted into the cell. That's not going to happen for free. That's moving in the wrong direction, right? If we simply open a gate allow the sugar molecules to move through, they're going to move from left to right, not from right to left. In order for this to happen, we're actually ending up with a higher state of energy, right? There's potential energy with a high concentration of sugar on the left, a low concentration on the right. If we move molecules, if we allow molecules to go from the left to the right, that would be taking us towards a lower energy state. But now we're actually saying we're moving molecules from the right to the left, we're taking it to a higher energy state, and that's why it's going to require energy. Okay? So I've covered this, I've covered that, the idea that we're having an endpoint with higher potential energy. That's kind of important. And in order for us to do that, the energy has to be coming from someplace. In this case, we're using a reaction that's going to be going to a lower state of free energy, right? Now one of those sources, source number one, might be a chemical conversion of a high energy molecule like ATP to low energy end products like ADP plus phosphate. Okay, so P subscript italicized I is usually the way that we depict the phosphate that you're cleaving off of ATP. Remember, ATP is adenosine triphosphate, ADP is adenosine diphosphate, tri means 3, di means 2, and so the extra phosphate is simply being cleaved off, right? So this is basically high energy on the left, low energy on the right, and so we're actually using the energy released from here to go into here to drive the movement of sugars from the right side, where they're at lower concentration, to the left side, where they're at higher concentration. Okay. Source number two, okay, might be a chemical gradient. We actually talked about this in the previous video, where we said that a concentration gradient where you have high concentration on one side, low concentration on the other side, right? This is actually stored energy, and when we move molecules from the left to the right, that's actually going to be an exergonic reaction. That's energy released. Okay. And so the two ideas that I want you to have here is that this process, this actual movement of a molecule or a solute from an area where it's at lower concentration to an area where it's at higher concentration, one, it does require an energy source. And two, that energy source may come from a chemical reaction. Usually that's going to be ATP being converted into ADP and phosphate. Or also pretty frequently, the source of that energy being used right here is coming from another gradient, a second gradient. Maybe something where we've got a high concentration of, maybe we have a high concentration, let's see, do this in green, a high concentration of sodium. Okay. And here we'll have a low concentration of sodium. And so maybe another way of driving this through is to say, well, okay, this protein here is going to attach one sugar, and it's also going to attach one sodium. Okay? And so the sodium is going to be pushing through, but it's also going to carry with it the sugar molecules that are going to be going in the direction that we need them to go. We're actually using the energy from this gradient to drive this transport of sugars across the membrane. Okay, and so I'm going to shift over to the textbook. I want to go to this spot in chapter 5, section 9. Again, this is something for the benefit of those who have access to the videos. If you don't have access to the video, I'm going to go ahead and run this and kind of talk you through it. There is narration. There's a really good narration that goes along but I'm not going to use the that narration. I'm going to use my own. Okay, so I'll be starting and stopping. We'll be talking about active transport here, and this is where we see the phospholipid bilayer molecules out here. Let's see if I can annotate. You can see the phospholipid bilayers. Annotate. Phospholipid bilayer here is a phospholipid. 2 layers. We've been talking about that a lot recently, and this would be an active transport molecule. And pay attention to this molecule right here. That's the one molecule that's got a lot of spikes on it. That's supposed to represent ATP. Okay. Now this molecule is going to be coming in. It'll be providing the energy to drive the movement of these solutes. Basically, this would be the solute that we're moving from the bottom to the top, right? And so after the ATP does its trick, it's going to be released in the form of ADP, which is the yellow ball without the spikes. The lack of spikes is intended to represent the loss of energy. Okay? So pay attention to this. So here come the two. Here come the two. Here come 3 sodium molecules they're coming in from the bottom. And we're also going to be having those move across, and potassium is going to be coming in from the top. Sorry, I misspoke there. Okay, so this is a sodium-potassium pump. 3 sodium ions are going from the bottom to the top. 2 potassium ions, the red diamonds, are going to go from the top to the bottom, and it's going to be driven by the ATP. Okay, see the sodium are going to the top, the ATP is being converted to ADP, and the potassiums now are going to be pushed down into the bottom. Okay. And this sodium ion potassium ion pump is one of the, this is actually one of the principal forms of transport that occurs in our cells as animals, right? We actually use this sodium potassium pump to create the gradients. For example, here, we know that potassium is moving down. In other words, potassium is going to be high down below, but I think more to the point, sodium is going to be high above. Yeah, draw that in. So draw sodium high. Those are the little yellow balls because we've been transporting them out of the cell. Potassium is high inside, but sodium is really low. Okay. And so we're actually using this gradient, sodium high outside the cell, sodium low inside the cell. The movement of sodium into the cell is going to be used to drive the import of the sugar molecule, right? These little tan hexagons are the molecules of sugar that are going to be transported into the cell using the sodium gradient. So let's see that. Continue on. Right? So glucose coming in, and we don't need to drive this with ATP energy because the sodium ions moving through is providing the energy that's used to move the glucose molecules into the cell. Okay, and so yeah, that was a video that I felt was worthwhile sharing. Okay, the last bit that we want to cover, and this is important because we're actually talking next about fueling reactions and energy metabolism. Okay. It requires us to talk a little bit about energy, fuel, and use almost like physical sciences terms. And we actually learn a little bit about thermodynamics here. And so the explanations here are really good. Please do read through this. I'm going to use a little bit of my own context and going back to my whiteboard to share. And so we've talked about ATP as an energy source, another source being concentration gradient. When you have a concentration gradient, we would call that co-transport. Right? Now, what we've been talking about, things like active transport or removing molecules from an area of low concentration to an area where there's higher concentration, it requires an energy source, right? And by definition, that makes active transport a form of work, right? So work in a strictly scientific context, I'm talking about what does a physical scientist consider work to be, okay? And that's simple. Any process that requires an energy input is work, right? So sitting idly in your chair, you might not think that you're working, but you are consuming energy, right? Keeping your posture upright requires your muscles to act. In order to do that, you have to have your muscles fed and you're having energy input occur. And so, yeah, you're actually working as you're sitting there completely idle, right? Not that that's going to help you at all in other contexts where work kind of requires you to actually do something more physical or more mental, right? Work, as we're talking about it here, is any process that requires energy input. And we've already talked about two examples now. Active transport very obviously requires energy input. Another thing we talked about earlier was anabolism. Remember, dehydration synthesis is something that happens only if we add energy in. Right? There has to be some type of energy source in order to take amino acids and put them together to form a polypeptide. Okay? The opposite, catabolism, involving hydrolysis. We end up with molecules that have a lower energy state than what we start off with, and therefore catabolism would not be an example of work. It would release energy, it would be exergonic, and therefore you wouldn't need to throw energy in, right? Anabolism is work. It requires energy, right? And so here we're actually talking about two classes of work. We would call active transport work, and we would call anabolism chemical work, right? This is actually physical sciences terms, chemistry terms. We're actually seeing things move from an area of low concentration to an area of higher concentration, requiring energy transport work. Converting these covalent bonds into those covalent bonds, and those covalent bonds represent a higher energy state, that would be chemical work, and anabolism is a good example of that. Okay? The third kind of work that we can relate to, maybe even better than the first two kinds of work, is mechanical work. Okay, and mechanical work also requires an input of energy, and but this is the kind of work that actually involves things moving around. Okay, so your movements, your physical movements, require the actions of muscles on your skeleton, and making all that happen is not going to happen for free. You have to throw energy at that, and that would be an example of mechanical work. Okay, now notice work is something that requires energy, but work is not energy itself, right? You know, there's something that's distinct from work and the energy that's required in order to drive the work process from going forward, right? So this is kind of like the hardest thing for a lot of beginning students, because you have these concepts that are really kind of related to each other, you know, work and energy, fuel, right? Are they the same thing? No, they're absolutely not the same thing. Right? So energy is maybe the most squirrely of all the concepts. It's something that you really can't put your finger on and touch, right? But you know that energy is being put in there because we're actually going to a higher energy state at the end, right? We know that a disaccharide is at a higher energy state relative to the two monosaccharides that are put together, right? And so we can touch the two monosaccharides, we can touch the disaccharide at the end, but in order to have the two monosaccharides undergo dehydration and synthesis to form the disaccharide, we have to throw energy in the process. So energy really is a thing, but it's not a physical thing. It's not a material thing. Right? Another good example is that sometimes we talk about ATP energy. Okay? ATP itself is not energy. Right? You could say that we're going to be driving active transport, we'll be driving active work by using the energy that's released when ATP is broken into ADP and phosphate. Right? This is the reaction. That's the exergonic reaction that's providing the energy. What energy is not there in the form of ADP and phosphate, but is there at ATP? That energy has now been transferred over to those molecules that have been put together in the anabolic reaction. Right? Got that? Okay. What would qualify as energy? I mean, apart from this kind of intangible thing that we can't, it kind of hurts your brain to think about, well, what is it that's going into an anabolic reaction? Well, energy. Can I see it? No. Can I feel it? Kind of hard to do that. But there are forms of energy that you can see and feel. Right? Light, for example, is a form of energy. Heat is a form of energy. And so these are more tangible things, maybe not tangible in a literal sense, but tangible in the sense that we can relate to them directly. Right? On the other hand, take something like gasoline. Is gasoline an example of energy? No, absolutely not. Gasoline is something you can actually put your finger on and touch. And so gasoline would be an example of matter. Okay? If it's material stuff, if there's a physical entity that you can touch, that would be matter, that would not be energy. ATP is matter. Gasoline is matter, right? But sometimes matter has a lot of potential energy stored in the form of relatively unstable covalent bonds, like, for example, disaccharide versus two monosaccharides. Right? Because, well, why is it that energy is required if we go in one direction, but energy is not required if we go in the other direction? Well, it's because the bonds in the two monosaccharides are somewhat more stable than the bonds that we have in the disaccharide and water. Right? And so if the energy stored in the bonds on the left side of the reaction is really pretty great, we would say there's a great deal of potential energy in those covalent bonds. The matter that has those covalent bonds would be considered fuel. Right? Here's our last buzzword of the day. Gasoline is obviously an example of fuel, right? ATP is an example of a fuel. This time you can actually see the bond that once hooked together the ADP and the phosphate. That was the unstable bond that is being reorganized in favor of more stable bonds. And that's where the energy is coming from when ATP is used by a cell, right? Okay. Okay, so what happens when ATP's bond, unstable bond, is reorganized? Well, energy is released. What happens to that energy? Well, it's going to go into some type of work, active transport work, chemical work like anabolism, or mechanical work, right? Now, take some time to think about, well, where are the differences between fuel, which is not the same as energy, which is not the same as work, which is not the same as fuel. Okay, these are three distinct things, right? They're all very real things. Try to sort out in your mind how they're different, because this is a really good exercise that brings your mind to a greater state of discipline. Right? That's it for this lecture.